So on that inverted section that we had put it for last for last class, right, for Monday, I have put on this PowerPoint the most important slides that you absolutely have to know. I know it's a lot of information in there, so I'm just going to fly through very briefly the stuff that you absolutely have to know, right? So on your inverted sessions, you will see, if you did not watch it yet, that you have different types of channels. They're responsive, right? So you have your chemically gated channels. That's the ones that you typically find in neuromuscular junction, right? And when you have transformation of electrical to chemical back to electrical, the chemical to electrical signal on the postsynaptic membrane, that's when you're going to find your chemically gated channels. So it's also called ligand gated. Besides that, you have the channels that are voltage gated. So those are the ones that feel the change in the electricity inside of the cell, right? And you also have those channels that are mechanically gated. So if they feel a little stretch or a little change in pressure, then they get activated. Okay. Things that we probably have not seen before on what is happening with your membrane potential, right, with your resting membrane potential. Sometimes you open channels like a sodium channel and you cause increase in the voltage that is inside of the cell. So that is called depolarization. So when you have something that goes up in the voltage scale, you are depolarizing the membrane. On the other hand, if you cause some channels to open and your membrane potential gets more negative, so going down, so you call that a hyperpolarization. You also call it an inhibitory signal. Okay? All right. What is the membrane potential and how you measure it? Basically, you are going to take into account the concentration of ions and other charged things that you have on the outside of the cell in relation to the things that you have on the inside of the cell. So we know that the sodium-potassium pump kicks 3 sodiums out of the cell and 2 potassiums into the cell, right? And that uses ATP. So when you look at the concentration of sodium on the outside of the cell, you will see that the sodium concentration is a lot higher than your sodium concentration on the inside of the cell, and vice versa for potassium, right? So you accumulate charges that are more positive on the outside of the cell in comparison to the charges that are inside of the cell. Now notice that you have these channels on the membrane. They're called leakage channels. So they leak ions out, mainly potassium. So you have some leakage potassium channels that allow some potassium to escape from the inside of the cell and to go to the outside. Okay? So if potassium is escaping the inside to go to the outside, so the inside of the cell will actually become more negative. Does that make sense? Hopefully. All right, so if you have something positive that is going out, then this is going to become more negative. Now, you need to take also in consideration what is the equilibrium constant for each particular ion. And this thing is not as simple as it sounds, okay? So you not only have to take in consideration the concentration, but also the electrostatic equilibrium of each ion. And we're not going to go into complicated equations of Nernst equation or anything like that. But I will mention a few things here, because this is a part that it gets a bit confusing, okay? So how do you measure the resting potential? You put an electrode outside of the cell, very close to membrane, and another electrode inside of the cell, very close to membrane. So the difference in the voltage is what you call the resting potential for that cell. So the inside is always going to be more negative because of the sodium-potassium pump and other reasons. Okay, so here is just one slide talking a little bit about the equilibrium potential of the membrane. So membrane equilibrium potential, okay? And what it says is that two things are important for dictating how much you're going to have of each ion on one side or on the other side of that partially permeable membrane, okay, selective membrane. So here, let's say that if you look at the sodium ions, like on the outside here, you're going to have more sodium ions on the outside and less sodium ions on the inside because of your sodium-potassium pump. So if you look at the concentration, you'll see that you have more here and less there. So the natural diffusion will make this thing to go inside of the cell. Okay. Now, if you look at charges differences, you have more negative charges inside of the cell. So it's going to also attract some of your positive ions, right? But then at the same time, you are going to have electrostatic interactions that are going to be counterintuitive or counteracting the concentration gradients. So I don't want to be elaborating too much of this because it's going to give me a headache. So mainly, you have to look at the overall equilibrium potential for each of the ions, and then you end up with a equilibrium constant. Okay, so also, no, don't worry about that. So main idea here is the following. When you look at equilibrium potentials for sodium, okay, you will see that when all of the concentrations and electrostatic interactions are taken into account and you open all of your sodium channels, the membrane potential is going to tend to go to the positive side. Okay, so here is 0. So if you have sodium and you open all the sodium channels, the equilibrium for sodium alone is going to be at positive 60 millivolts. Okay? If you want to do all the chemistry and figure out why that's the number, be my guest. I'm not going to say anything about that. Okay. So with all the sodium channels open, you're going to be at a positive range. Okay? Now, forget about sodium. If you open all of the potassium gates, what is going to happen with potassium, if you open all the channels, potassium is going to go to the outside of the cell, and the membrane potential for potassium is going to be very negative. The equilibrium potential for potassium is going to be around negative 90 millivolts. That's going to be the measurement of the membrane potential if all the potassium channels are open. Okay. And then if you forget also about potassium and we look at your chloride ions here, if you open all of the chloride ions, they will tend to go to the inside of the cell, okay? And the equilibrium for chloride ions is going to be negative 70. It's also going to be very much lower. It's going to be negative, okay? But then notice here that the equilibrium for chloride ions is actually very close to the resting membrane potential, okay? So that is interesting because when you think about what is causing the resting membrane potential, that is sodium-potassium pump. So chloride ions are not involved in maintaining resting membrane potential. So the resting membrane potential is not going to really influence a change for that resting membrane potential, right? So looking at them individually, they will tend to bring the membrane to different electrical resting potentials. All right. But then when you start your sodium potassium pump again, everything is going to be equilibrated at a negative 70 millivolts. Okay. So here comes your first question. Okay. Which type of membrane channel opens only when a specific chemical, such as neurotransmitter or a ligand, binds to it? I already gave you the answer. I don't think you need more than 30 seconds to answer that. Does anybody need more time? No? You're going to stop it? What is your answer? C. That is your chemically gated channel. Okay. Okay. In case you would have all the potassium channels open, right, the resting potential would be very, very much negative, right? But you don't have all of those open. You're going to have them closed, and you're going to have sodium potassium pump working, so your resting membrane potential is not going to be that low. Okay. Now, here is what is going to happen in case you have only sodium channels open. Then your inside of the cell membrane potential will be very positive, but you know that's not really what happens because of that low permeability for sodium and the sodium potassium pump working. So here is what happens when you look at the action potential. You have a certain membrane potential and disturbance of the membrane potential, that if you reach a certain threshold, you're going to cause this all or none type of permeability changes and propagation of your membrane potential. All right. So here is a graph showing the permeability of the ions, also together with the membrane potential. So here we look at this blue line that says sodium permeability. So you see that the permeability of sodium is going to sharply increase once you reach that threshold. Okay, so the red one is for the threshold. If you reach threshold, your permeability shoots up. But when you reach inside membrane potential that is close to positive 30 millivolts, remember what happens to your sodium channels? They inactivate. If they inactivate, that means you totally cut the permeability of sodium. So when you inactivate, what happens to permeability? It plummets down to almost no permeability. Okay? So here is the period of time when your sodium channel is open. Now, after your sodium channel inactivates, this is when your potassium channel opens. So here is your permeability for potassium. Notice that the permeability for potassium is a bit higher than the resting permeability for sodium, because potassium has those leakage channels, right? Just leakage channels. So after you inactivate sodium, this is when potassium channels will open, and they will also close and remain closed until your next action potential. Right? So make sure you understand what this graph is telling you about the action potential and about these different permeabilities for the ions. When you are asked what is the magnitude of an action potential, you have to look at what was the resting potential, or what state was your cell before you started the action potential. So you have to look at the beginning of your action potential up to how high it got. So this is going to give you the magnitude of your action potential. Okay, that is the magnitude of the action potential. If you started for normal resting membrane, So you just do the addition on absolute values, and you get your action potential magnitude. You know what I'm talking about, absolute values? Yes? Everybody took math? Hopefully. Okay, so what is your magnitude here? 100. Everybody got 100? Yeah, okay. Now, look that you can actually have magnitudes that could be higher than 100 in case you got to that relative refractory period, right? In which your sodium channels are closed but not inactivated. And your membrane potential is not back to normal resting potential, and you are a bit on the hyperpolarized part, right? So here, if you put enough signal, if you send enough of an impulse, you could reach threshold, and in that case, your magnitude would be like from -80, up to 30. So then your magnitude would be higher, right? Would be like 110. Okay, so if you are at the relative refractory period, you could send a strong signal and cause action potential to happen. Okay, so you should know about your permeabilities, right? So influx of what and influx of what else? Okay. Okay, so this observation is important. So all of the action potentials, they are about the same. Okay, they are alike. And what you see that is different with neurons is that you can have inputs at the dendrites that may or may not lead to the triggering of action potentials. Okay. So let's see a bit more on that. Okay. So here, if you are sending a stimulus that is weak, so you're sending stimulus through dendrites and is not enough to reach threshold at the axon hillock, so then you do not get action potential. So if you do not reach threshold, you don't get any action potential. But let's say that you send a strong signal and you reach threshold for quite some time. So if your signal is strong enough to cause action potentials, and not only one but several of them, so you are going to have your action potentials. They are going to be firing, and depending on how strong your signal is, that is going to tell what is the frequency of your firing for the action potentials. But if you look at the magnitude of each individual action potential, that is going to be the same. Okay. Now, if you send a very strong signal, a stronger signal, then you can actually fire more action potentials sequentially, but then each one individually is equivalent. So make sure you understand what that means. Okay? Signal strength in relation to firing of action potentials. Okay. So this part that says how fast is an action potential going to travel down the axon? Okay. That is going to depend on a couple of things. First is going to depend on the diameter, or how thick the axon is. It's also going to depend on whether your axon is myelinated or not. And it's also going to depend on how long your axon is. Okay, but those two factors here are the most important factors to determine how quickly the action potential is going to reach the other side. Okay, so what is the relationship? The bigger the diameter, the less resistance you're going to have for your action potential to travel. Okay, so the bigger the diameter, the faster the action potential will travel. Now here, concerning myelination, if you have myelination, you're even going to have faster transmission of the action potential. Okay? So if you have myelination, you're going to have what you call saltatory conduction. So the saltatory conduction is going to go from node of Ranvier to another node of Ranvier, which I had mentioned before. So this is myelinated saltatory conduction. If you do not have myelination, then you're going to have this continuous conduction, and that tends to use more energy and it tends to be slower. And you have other problems that are also involved with that. So here is a visual showing in comparison of diameter of the axon. So here you have your action potential going a bit slower than your other action potential if you start firing all of those at the same time. Okay, so skinny axon, you go slowly. Thicker axon, you go faster. Myelinated axon, you go a lot faster, even if your axon is a lot skinnier than that axon over there. Okay, so myelination makes it go a lot faster. All right, so here is another question. Okay. Which of the following increases the speed of action potential along the axon? So you want to have a very fast action potential. Which one did you choose here? Does anybody need more time? No? Everybody got your points? Yeah, okay, so I'm going to stop it. What is your answer? C. Okay, so there's going to be large axon diameter and myelinated. All right. So now we get to the fun part of graded potentials. So those are the potentials that are happening at the dendrites, not at the axon. Okay. When we're talking about axon, action potential, once you passed this initial firing at the axon hillock, then this is going to be the all or none type of conduction. But when you're looking here at your dendrites, is receiving, those are receiving information. Now, this is when you can have decision making on whether you're going to fire, do you have enough excitatory or do you have more inhibitory. So a lot of stuff is getting processed over here before you make a decision of firing or not. So those are the graded potentials that we're talking about. So this really has to do with this part of the neuron that is receiving the information. Okay. How is it that the dendrites are going to receive the information? Well, there are a couple of ways. There are several ways they can receive information. They can receive a lot of information in series. So if you get one dendrite here, you can send one information, and then you send another signal, and then another signal. So those in series, you call it a temporal type of summation. So you do like, you do one, and then you can have a little time before the next one on the same dendrite. You wait a little bit more, get the next one on the same dendrite. So this one is your temporal summation. You're going to sum them all up, and if you get enough signal that is going to trigger the action potential here at the hillock, then you're going to get your firing down the axon. Then you're going to get that, all or none type of response. Now, if instead of that, you have information that is coming at the same time at different dendrites, that also gets summed up, but in this case, you're spreading your stimulus at the same time through a bigger space, so you call that spatial type of summation of these graded potentials. So here, when you compare the graded potentials with the action potential, right? So you have to know how they're different. Okay. So for the action potential, remember that's the all or none going down the axon. This action potential has that positive feedback characteristics because of the opening of the channels. Once the channels feel the change of the resting membrane, okay, or of the membrane potential, I mean. So we looked at the characteristics of the action potential, right? When you compare it with the different types of potentials that you can have at the dendrites, then the situation is different. So you see here that you can have all different sorts of potentials that are coming through the dendrites. They can be tiny changes on the membrane, they can be bigger changes on the membrane. So this is what characterizes them as graded. So you can have a little bit, you can have bigger ones, so they're not all the same size. Okay. Now, if you look at where the signal started, and then you look at the other side of the cell, you can see that this signal actually faded away. So it changes or it decays the farther away from the receptive point. Okay, so it can fade away with the distance. Now, what kind of signals that are going to be causing changes in this membrane polarity? Basically, you can have all kinds of signals. You can have, chemical signals or stretchy signals, right? So it's not just electrical signal in comparison to the action potential that is only electrical, right? Also, in these signals here, you do not have that positive feedback. That positive feedback would give the characteristic of all or none, and these guys don't have the all or none business, okay? And also the repolarization at the membrane is not really dependent on the voltage right after that graded potential. Okay, so those are the comparisons on how you distinguish what is a graded potential and where it's happening in comparison to the action potential. Okay. So here, looking at those graded potentials, right? So those are here at the receiving side of those dendrites. So you can have two types of inputs, right? Remember, you could have two ways of putting the input. Either you have the temporal, or you have the spatial, right? And here you can have excitatory signals, and you can also have inhibitory signals. All right. So what is it that's going to give you an excitatory signal? Basically, you're going to look at depolarization of your membrane. So you have to look at the ions, and you're going to say which ion is going to cause depolarization of the membrane. So mainly you're going to see that sodium is the very guilty one, is the responsible one for depolarizing the membrane. But then when you have sodium channel opening, you are also going to have potassium channel opening. But remember, potassium is going to cause repolarization of the membrane, right? But these guys here are supposed to be excitatory. So you mainly depend on your sodium channels, okay? Because you want to get depolarization of the membrane. So here is showing your sodium, okay, your sodium channel. And here is showing you a slight depolarization of the membrane. All right, those are your excitatory ones. When you look at inhibitory signals, you are going to look at ions that if you open the channel, the inside of the cell is going to become more negative. And those are going to be potassium and also chloride channels. Okay, so when you open potassium channels, potassium is going to fly out from the inside of the cell, and the inside of the cell is going to become more negative. So opening only potassium channel is going to cause an inhibitory signal, and that is called a hyperpolarization. Okay, it could also cause a hyperpolarization. When you have both of these opening at the same time, then you have to really do the summation and see which one is winning the race. Okay, when you open your chloride channels, chloride tends to be accumulated on the outside of the cell, so it has a tendency of going to the inside of the cell. So here is something negative that goes to the inside of the cell. You are going to hyperpolarize your membrane. All right, so those are the examples, those are the cases in which you can get inhibitory signals. And again, everything is happening here at the dendrites. Everybody is clear with that? Yeah, all right. Okay. So here, what is happening here? When you have excitatory signals, right, you're going to have that influx of sodium and also potassium, but then sodium wins. And here is the explanation, so you can read that on your own. This guy here wins over the potassium, and that thing is excitatory, and you might get to threshold or not. but you're going to be adding up your excitatory signals to see if you're going to get to threshold or not. And this threshold is really being measured at the axon hillock, because everything else that is happening over here is only getting summed up. What really counts is what's happening at the axon hillock, because that's what's going to decide whether you're going to fire that action potential down the axon or not. Okay. In comparison here to your inhibitory signals, again, what is governing inhibitory signals are those guys, and you're going to have hyperpolarization, and it's going to make it more difficult for that neuron to fire. Right. So here is again telling you about the idea of adding up all of those signals that are getting to the receiving end of the neuron, right? So you call it summation, right? So you summate or you add them together, right? And again, you can have either temporal or spatial summation. So here is the idea of this neuron on a rocking chair. So he's rocking, every time he's rocking, he's going fire, fire, fire. It's not very convinced to fire, right? But it is adding it up. It is almost getting an action potential, but it's in series. So it takes a little time from one signal to the next. That's your temporal. And here, when you get your spatial summation, so now let's say that you're getting a lot of people that, oh, this guy's not in the rocking chair anymore, right? So let's say you get a lot of people now that get together close to this guy. Let's assume it was the neuron was asleep, and then you get 4 people that goes at the same time on the side of the bed, and the guy goes, fire! So now this guy is going to jump up like, what the hell, right? So it's not on the rocking chair, and fire, fire. No, it's like I wrote ones like 4, like fire. So now, yeah, you can get to your threshold, you know, a lot quicker when you get this spatial summation of this poor guy over here. Okay. And now let's say that you have some people that are actually confused. You have 2 neurons that are sending signals to, let's draw another one here. Oh, here they are. Okay, so 2 neurons here sending signal to this guy over here, and one neuron is going fire, fire, and this guy's like, okay, I'm thinking about firing, but the other neuron here is going, don't fire, don't fire. So this poor neuron receiving the signals is going like, should I fire, should I not fire, should I fire, should I not fire? So it's not quite reaching threshold because it's having conflicting information getting to that neuron at the same time. And yet all this information is getting summated to decide whether the thing is going to fire or not. Okay. Oh, another. Which type of potential does not decay with the distance? What's a generator? Oh, thank you. Somebody gave up the generator. So you should really have like 50% chance of getting this right. But if you are awake and neurons in your brain are going like fire, 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 you should get the correct answer, right? Anybody need more time? Yes, the ones that are giving me the wrong answer, you should consult your friends quickly. Okay, what's your answer? B, the one that does not decay with distance is your action potential. Okay. And finally, we're going to get to the brain and the spinal cord. Everybody ready for the brain? Okay, so here we go. Brain and spinal cord. Things that you absolutely have to know is, again, the type of neurons, right? Because they're important for a brain and spinal cord. So here's a little slide doing a bit of a summary on the neuron types, okay? And where you find them. And your supporting cells, your glial cells, neuroglia, right? So you have here your microglial cells and what they do. You know, they migrate, they eat up stuff. and you have your oligodendrocytes doing the myelin sheaths, and you have your astrocytes that are very important for blood-brain barrier, right? So just quick review. All right. And here we get to the development of the brain. Right? So when you start with the egg and the sperm, you're going to have cell division, and then you're going to start having those different layers of development, morula, blastula, And here is when you're going to have your ectoderm, that is the tissue that is going to make your skin and also your nervous system. And you're going to have also mesoderm. You don't even have the endoderm over here. But basically, you're going to be focusing here on your ectoderm. So you're going to have a little indentation of an area of the ectoderm in which you call the neural plate. Then this is where you're going to start having the development of the neural tube. Okay, so this thing goes down and it makes a little concave formation, right? It does this convergence thing. You call this a neural crest. And then you're going to have a fusion of the ectoderm and the formation of this neural tube. Okay, so everything here really came from your ectoderm. All right. So what is happening here with the brain development? So when you have that neural tube formation, so here you have your central or neural canal that is being formed. So in this case here, we just flipped the image upside down. So on the other slide, we have the tube going this way. So here we have the tube going that way. Okay. So what is going to happen here? You have three areas that are forming like different little holes that you call vesicles, okay, like baskets. So the first area over here that you call prosencephalon, they form this first little area of vesicle here. This is what's going to make the forebrain. The middle section, you call it mesencephalon, and that is going to make your midbrain. And then the bottom section, you call it the rhombencephalon, and that is going to make the hindbrain. So this is the beginning of the development of the brain, also with the spinal cord. And this is before you get further subdivision. So that's the beginning stage, 3-stage of vesicle. Okay. After that, you're going to get to the 5 vesicle stages. So the first division that we had here is going to subdivide into 2. So you're going to have here your telencephalon and your diencephalon. Okay. The mesencephalon is going to be the same. Okay. And now you're going to have also the bottom part that is going to divide into 2 other sections. So you have metencephalon and myelencephalon. Okay. So the top part and the bottom part got subdivided. The middle stayed the same. All right. And the thing keeps on growing, right? So at this point, you're going to have further differentiation of those subcompartments, and you're going to start having more characteristic shapes of what this thing is going to become, right? When you look here at the upper portion, you're going to find that you're going to have this indentation on both sides. They're going to be specific ventricles. They're going to make spaces that you're going to have fluids in them. So you're going to have 2 ventricles over here. So you're going to copy 1 and 2. So those are your lateral ventricles. And then the one that goes in the middle, you call that your 3rd ventricle. Now, the space between the first 3 ventricles and the other space, this is not really a ventricle. This is just a little passageway. So you don't call that a ventricle. Ventricle is something where you can keep the liquid in there, like store it in there. This thing is only a passageway. So you call that an aqueduct. It's going to allow liquid to go through, okay, like a straw kind of thing. Okay, so this is like an aqueduct. And then your 4th ventricle is going to be in this area over here, okay? That's going to be close to cerebellum and the pons at the hindbrain. Okay, so midbrain, you're going to have the aqueduct, and then hindbrain, you're going to have your 4th ventricle. And the forebrain, you're going to have 3 main ventricles, two lateral and one in the center. Okay, so what is important to know here? Okay, so your metencephalon, that one is going to become the pons and cerebellum, and we're going to see more on where those are located and what they look like, right? The myelencephalon is going to become the medulla, okay, medulla oblongata, and then is going to become the spinal cord. All right, so this is from your book showing the developmental stages and which structures they develop into. And again, you can read that on your own. Okay, so let's get you more interesting things. Oh, okay. Another question. The cerebral cortex arises from which type of secondary brain vesicle? This one you have a good mnemonic to help you out. Like when you go like this, I'm going to give you the idea by telepathy, right? You're thinking about your... No, nobody should give me the wrong answer. Using your brain, your cerebral cortex. Anybody need more time? Okay, what is your answer? Hey, your telencephalon. Come on, okay, here we go. All right. So here, looking at the whole brain after it has developed, right? So you're going to look at two halves. So you're going to have the hemispheres, and of course here we don't see because it's looking at the lateral side, right? So you're going to have two halves. And here, looking at the whole brain, you're going to have the cerebrum is a bigger part, right? And here you're going to have your diencephalon, that is the more inside middle area of the brain. And this area is very important for regions that control a lot of body functions and also memory formation. Okay, so here we're going to have thalamus and the hypothalamus that is right in the center, that is close here to the pituitary gland, okay, and epithalamus. This is all going to be this diencephalon purple area here. Then you look a little bit under and also central, you're going to have your midbrain. So all this green part here is going to be the midbrain. Now, what is the midbrain going to have? Here, you're going to have your brainstem, okay, which is right there. This is all part of your brainstem right here. In the brainstem, you're going to have the midbrain. You're going to have the pons right there. And here is the medulla oblongata that is going to connect this midbrain to the spinal cord. So spinal cord is going to be down here, right? So diencephalon in the middle and cerebrum, that's the big part over there, right? Now, this is what we are showing in here. And on the back, this brown section here, you're going to have your cerebellum. And it's actually, it is very easy to distinguish the cerebellum from your cerebrum. Let's look more into that. Oh, another one. Lots of points today. The pons belong to which part of the brain? So you got to be familiar with where things are located in the brain, right? And what you call them. It's funny, I always get somebody who answers E. Good, they change their minds. Okay. Does anyone need more time? Everybody answered? Okay, what is your answer? D. That is your brain stem. Okay. So now let's look at these ventricles at the mature brain. Okay, so ventricles are going to have fluid in them, and the fluid is your cerebrospinal fluid. Now we're going to look at the cells that are responsible for making that cerebrospinal fluid, and we have briefly seen before the cells that are moving the cerebrospinal fluid around. You remember what those cells are? The pindinal cells. Okay, those have the cilia, they move the stuff around. Okay. So now looking at these ventricles over here, you see that there are two ventricles that are part of each of the hemispheres right in the center. So here is ventricle 1, ventricle 2, those are your lateral ventricles. And then if you look right in the middle of this 1 and 2, you're going to find your third ventricle, right? And after that, you're going to find that that area that connects the ventricles, that is not really a ventricle, so that is your aqueduct. And then after that, you're going to have here your fourth ventricle. Okay, so make sure you know where they are. Okay, and here, looking inside of the ventricles, what you're going to see? You're going to have here for the first and second, you're going to have this shape that looks like, I don't know, a screaming butterfly. So you have here butterfly antennas and butterfly wings, right? And you don't need to know exactly which area is where. You can just read for your enjoyment. But you should know that this is first ventricle and then second ventricle. And then here you have your third ventricle. And here is your aqueduct, and here you go to your fourth ventricle. Now, important thing is that you have to know here is that you have these areas that are in red, okay, and those areas are called choroid plexus. These areas have a lot of capillaries, a lot of little blood vessels. That you know that blood doesn't really get into your cerebrospinal fluid. The spinal fluid is supposed to be clear, right? But the clear thing that is coming to those ventricles, those are coming from the plasma of those of the blood that is going through those capillaries. Okay, so this is your choroid plexus that is really supplying the cerebrospinal fluid that is going to fill up those ventricles. Now here you have the spinal fluid that's coming from the choroid plexus, and you see here that in the fourth ventricle you're actually going to have a drainage system for the cerebrospinal fluid. So here you're going to have circulation of the fluid everywhere, and around the fourth ventricle you're going to have some drainage of the spinal fluid that is going to allow the fluid to go into the meninges space and to cushion the outside of the brain. So this is the inside of the brain that gets some cushioning, and then on the outside of the brain you're going to have another level of cerebrospinal fluid. Okay? Okay. In case you have a problem with the draining at your fourth ventricle and the cerebrospinal fluid is not quite draining out, that can cause a big problem, right? If that happens during formation of the fetus, this is when one of the cases in which you can get hydrocephaly. Okay, so you see this baby here with this very big head, and if you look at coronal section of that brain, you'll see that the ventricles are huge. You barely have brain mass, and you have these huge, huge ventricles in the middle. So what happens here is that without draining the fluid out, fluid is going to accumulate in the ventricles. And because the heads of these babies, they are not fused yet, the bones are not fused and they're quite malleable, so you can have extension of the head and is expansion because it has to accommodate for these huge ventricles, right? So this is a case of hydrocephaly. Another problem that can cause that is not only the lack of drainage, but also overproduction of cerebrospinal fluid at the choroid plexus. okay, or then a combination of both. And what do you do when you see a patient that is born with this problem? What is the kind of treatment that that can be done? Draining, yeah, kind of draining, but not quite draining. Draining would be more like to the outside. What they do is actually the doctors will implant a shunt system that connects the ventricle, actually the fourth ventricle here, with the venous system. So it will be draining, but to the inside of the body, going to the venous circulation and back to the blood circulation. So slowly, this extra fluid gets drained into the circulation. Now you could be asking, like, what happens to the head of the baby? Well, that is when the doctors also design a helmet to allow control of that growth, or temporarily stop that growth of the cranium, right? So the rest of the baby will actually grow into the head. It's not going to shrink the head, right? That's not a problem. But if you control the growth and you allow drainage, then the child can recover and have a close to normal life. Okay. All right. All right, so now let's see what happens to the organization of neurons when you look in the brain. You'll see that you have an area that is close to the surface of the brain that you call the gray matter, right? So we're all using the gray matter, yeah? And you look deeper, you're going to see the white matter. So what is making up the gray matter and the white matter? When you look at the gray matter, you're going to find cell bodies and dendrites, okay? When you look at the white matter, you're mainly going to find myelinated axons. So if you're looking at something gray, you're looking at cell bodies. If you're looking at something white, you're looking at myelinated axons. But that subdivision, the gray is close to the outside, that is for the brain. Now, when you look at the spinal cord, that pattern shifts. So in the spinal cord, you're going to have the cell bodies and dendrites. They're going to be close to the center of the spinal cord, this section. And then the white matter is actually going to be on the outside part, on the periphery of the spinal cord. Okay, so here you have a shift of the white and the gray matter in comparison to the brain. Okay. Now, if you look again at the brain, you're going to see that inside of the brain, you're going to have some concentrations of gray matters. And those are going to be also bodies and dendrites. They're concentrated. And those are the nuclei, different nuclei. Nuclei, actually. Nucleus is one, nuclei is plural. All right. All right, so here is just what I have just mentioned, right? So gray matter is full of cell bodies and dendrites. White matter is full of myelinated axons. Okay. That's just a repeat with a pretty picture. Okay, I just mentioned that. And this is from your book. That is showing, again, your ventricles. Okay, so lateral ventricles, that's 1 and 2. This is a coronal section, right? So 1 and 2. So here you see more details on those nuclei. So you have gray matter here and gray matter there. So gray matter on the cortex. So that is really showing where you have your bodies and dendrites. So here is interesting when you look at the formation of the spinal cord, right? So very, very close to the brain, you still have your cortex that is made of gray matter. And notice that when you come out of that fourth ventricle, you have that ventricle that is extending to your medulla, okay, to the spinal cord, okay? So this ventricle here is surrounded by gray matter, Okay, but as you go down in the spinal cord, that opening becomes smaller. So the ventricle is called the central cavity, and still you're having cerebrospinal fluid that is going through that, right? And still you're having some gray matter on the surrounding. Now look at this. When you get down, further down the spinal column, you see that the central cavity becomes more like a very tiny opening. Okay, so your central cavity here is barely noticeable, and you notice that the gray matter actually had a distribution shift from being around the cavity to this butterfly shape. Okay, so here is at the cerebellum, here is at the brainstem, and then here more down at the spinal cord. Okay. All right, so how do you protect the brain? Because you know that's the most valuable organ that we have, right? You actually have at least 4 levels of protection for the brain. Okay, so the first one that we see here is, of course, the skull. So you have very dense bones that are going to be protecting the brain. Okay. Besides that, right under the skull, you're going to have the meninges, and yes, you have to know the meninges, right? And the meninges are also area that is going to protect the brain. And meninges are going to be bathed in cerebrospinal fluid, right? That's also going to be protective, right? So let's go like one by one here. So you have your dense bones. Then here you're going to have your meninges, right? There are 3 meninges. The one that is closest to the outside is your tough mother, right? So that's the dura mater. The one that is in the middle is the spidery mother. So here's your arachnoid mother, right in the middle. And the one that is the thinnest and it comes in contact with the brain tissue, that's your thin mother. Okay, so that's your pia mother. All right. So this is your second level of protection. Okay, the first one was just bones, right? First one the bone. Second level of protection is your meninges. There. Okay. And third level of protection is going to be your cerebrospinal fluid. So that is going to allow the brain to be like floating in that space. So it gives you some buoyancy and also shock absorbing session, right? So you're going to have that extra protection in case you hit your head. You have this liquid that kind of serves as a shock absorbing fluid. Okay. Again, what is it that is going to make your spinal fluid? Is going to be a bunch of capillaries, and those are your choroid plexus, as well as with your ependymal cells. Okay. And here is your fourth level of protection, that is the blood-brain barrier, right? So you have certain cells that go around the arteries or the capillaries, and they're going to be very important at selecting what is allowed to get to the brain, and most things are not going to get to the brain. So a couple of things that are very characteristic of nutrients that will go to the brain. One of them is glucose. So the brain has its own glucose transporters, right? So glucose will get to the brain because the brain likes to use glucose to work, right? But also lipids will get into the brain, okay? And a lot of times the brain works even better using lipids than it does using glucose, right? Have you heard of the ketogenic diet? Ketone bodies? Yeah, ketone bodies are very good for brain function. The brain gets happy with ketone bodies. But then it also gets happy with sugar. So, oh well, gotta keep your brain happy. So, cerebrospinal fluid, the blood-brain barrier is what does the selective barrier. Right? So this extra level of protection. Right. So organization of the brain, more here on the organization of the brain. You're going to have 2 hemispheres. The encephalon, we already saw that. Oh, I think that's a repeat. Okay, let's go ahead. All right. So we're going to see here on the different lobes and where they're located, right? And division of how to study the brain. The part in the front of the brain, that is your anterior portion. The back of the brain, of course, is the posterior portion. Okay, so looking in the front of the brain from the top, you're going to see that you have two halves. So here's going to be your left hemisphere, and here's going to be your right hemisphere. So two halves for your cerebrum. Okay, and now comes the fun stuff, right? Landmarks of the brain, or overview of the anatomy. So if you have things that are bulging, we call those gyrus, right? Not a sandwich, a bulging sandwich, or gyrus, okay? Plural is gyri. If you have things that are going inside, you call one a sulcus, right? More of those are sulci, right? So gyrus and sulcus. And then you're going to have some areas that they really are like dividing the brain into sections. So those are fissures. Okay, sulcus is a little bit of a division, like, but then these fissures are really very pronounced indentations. So what you should know here, the longitudinal fissure, that's the one that divides the left and the right brain. And then you're gonna have here your other fissure that is not showing the name here. Central, it should be in the next page. Oh, no, it's still on the following one. Okay, so longitudinal fissure is going to divide left and right, and then the very prominent fissure on the side, you call it the transverse. Where is the transverse fissure? It's still not very evident here, but you should know those two very characteristic fissures. Okay, fissures. So different lobes of the brain. So you have lobes and sulci and fissures that are very much landmarks. In the front of the brain, you're going to have your frontal lobe. Close to your ears, you're going to have here your temporal lobes. And after the frontal lobe, you're going to have a parietal lobe and your occipital lobe. And you can just match that with the bones of the skull, and you can very easily situate yourself. And here you're also going to have another area that is very prominent, that is your cerebellum. And that is termed cerebellum because it looks like a little brain. So it's kind of like a miniature brain close to the big brain. That's your cerebellum. And it's very important for movement and coordination, right? After the cerebellum, coming from the central brain, you're going to have your brainstem. Okay. So you're going to have 5 lobes that you should know. Right, so let's look at them here. Frontal lobe, parietal lobe, temporal lobe, and occipital lobe. And you should know the main functions of some of those, right? Frontal lobe is very important for thinking and reasoning, also for memory, right? And here parietal lobe is also important for a lot of reasoning and connections. But interesting here, when you have a junction here of your frontal lobe with your parietal lobe, you have two gyrus sessions that are very important. This one in the frontal lobe is responsible for all of the somatic coordination in the body. So voluntary muscles, voluntary movements will come from this gyrus that is right at the back of your frontal lobe, just before your parietal lobe. And then when you jump to the parietal lobe, you're going to have here this post-central gyrus that is going to be responsible for sensations of feelings, feeling of things. Okay? So when you look here at the occipital lobe, that's very much important for vision processing. And here when you look at temporal lobe, that's important for speech and hearing. Okay. Okay, so here we look at those, and we miss the fifth lobe that is called the insula. the insula is very much insulated inside of the brain. So you have to pull apart your frontal lobe and your temporal lobe to find the insula right there under it. Okay, so that's your fifth lobe. Okay. So here we're going to start getting into functional areas. Okay. So we know that the cerebral cortex, that's your gray matter, right, is very much important for reasoning, thinking, everything that your, decision making, that anything you can think of is happening at the cortex, but it's actually a very thin area that is very much superficial. That is your layer of your gray matter. Okay. Yet when you look at the gyrus and sulci, you see that the surface area is huge. So this cerebral cortex, although it's very thin, it makes up about 40% of the mass of the brain. And this is where you're going to have your conscious mind and all of the senses that are going to be processed. Okay. One last thing about this slide. The left side of the brain coordinates things that are on the right side of your body. The right side of the brain coordinates things that are on the left side of the body, and you call that contralateral type of coordination. All right, so I'm going to stop here, but remember, I'm going to put those videos for flipped classroom, because we need to catch up and finish up the topics. All right?